So now we're going to the next session, which is all online. So it's on Rhizodeposition and carbon storage in soils with Christine Hatté and Wafa Achouak and Thierry Heulin du BIAM.
Hello, everybody. I would like to share my screen if it is possible. Can you see my presentation?
Yes. Yeah. No.
Good. Thank you.
Non, c'est pas bon. On est en mode présentateur.
On est en mode présentateur. Mais moi, je suis jamais en mode présentateur.
Je pense que c'est le mode de partage. Il ne faut pas choisir. Faut sortir du mode de partage et re-rentrer pour choisir un autre mode.
Sur l'écran.
If she right click, Wafa, si tu mets dans présentation, un peu clique la souris à la droite et propose des choix. Clique à droite.
Clic droit.
Clique droit, le souris. Et te propose normalement un mode de présentateur ou un mode de speaker.
En fait, c'est avec Zoom, en partageant, tu as le choix entre PowerPoint ou l'écran, écran 1, écran 2 parfois.
J'ai qu'un seul écran.
Sinon, on partage ici et on fait next.
Je peux aussi partager depuis ce que j'ai sur mon écran si tu. Mais j'ai pas la dernière correction que tu as voulu implémenter.
Il y a deux écrans.
Vas-y, fais-le parce que je pourrais.
Il a des bureaux sur son Mac.
Moi, c'est possible que ça fasse pareil, dites-moi. C'est OK?
Ah oui, j'avais changé en anglais. So good. Everybody, so with Christine and Thierry, we'll take you into the rhizosphere with 3 voice presentation on the rhizodeposition and sequestration of carbon into soils. Next. So there are some limitation approaches to restoring organic matter to soils. Ça a changé, Christine? So agroforestry could be a solution. However, there are some constraints and and simply spreading crop residue such as effluent or compost only enriches the soil surface and can sometimes lead to mineralization of the existing carbon. So we question here whether varietal selection could be an alternative to conventional practices for carbon storage into soil. Next, please. So as you may know, plants invest up to 40% of what they produce of their photosynthetates as root exudates. It's a way for the plant to recruit a specific microbiota from the reservoir, which is the soil. And in response to some stresses, the plant may select and recruit another microbiota, which contribute to help the plant to tolerate some stresses. So some bacteria in the soil transform these root exudates into exopolymers, like exopolysaccharides. And these exopolysaccharides, they form organo-mineral complexes that we can see here. I don't know if you can see my cursor. And this complexes the modified the soil structure and also the porosity of the soil, improving the water and nutrient uptake by the plants. And also these polymers contribute to increase the size of this rhizosheet, this soil, this root adherent soil. And as it contains and maintains water, it retains water, it's a way for the plant to tolerate also water stress, because it can get uptake this water from this rhizosheet. So next, please. In the frame of an INR project, we, with colleagues from ERD, Laurent Cournac and Laurent Laplace, we used this rhizosheet as a proxy, and we screened 180 per millitumbrid lines based on this ratio on the root adherent soil on the root tissue. And as you can see here, when we compare the different per millet lines, we have great variation in this ratio with plants that have a large rhizosheet. So we selected lines that we consider here as low aggregated lines on the left, and two other lines that we consider as high aggregated lines in the right part. Next, please. And we aim here to determine if there is any correlation between the rhizosheet size, root exudate composition, and microbial community assemblage, and also if there is a difference in carbon storage in soil between these hybrid lines. Next, please. Next. So our colleagues from ERD, they used a GWAS genome-wide association analysis approach to analyze this contrasted hybrid lines. They showed that the rhizosheet size is genetically controlled, which may open a way for varietal selection, where this root trait could be considered. Next, please. So they mainly showed that there were differences in genes involved in the root exudation. So we established, no, please, no. We established an in situ system using native soil, and we employed a sensitive and targeted analytical approach, which is FTICR-MS with our colleagues from Helmholtz Zentrum in Germany. And we could identify compounds, different compounds that provide from root exudates or microbial metabolites that were activated by root exudates. And as you can see here, we characterized this metabolism of root exudates, of root adherent soil, which correspond to root exudates. And we can see a difference between low aggregated lines and high aggregated lines. And we could see these differences also in the root compartment and in the shoots. And we could identify some compounds that were specific to high aggregated lines and others that were specific to low aggregated lines, especially in the root adherent soil, which may explain, next please, the differences that we observe in the bacterial composition or fungal composition. That was the difference that we cannot see here now. So we used metabarcoding to identify bacterial populations in the right, in the left figure here. And we characterize bacterial population in the bulk soil, in the root adherent soil, and also in the root compartment. And we can see here a clear difference between high aggregated lines and low aggregated lines, especially in the root compartment. We observed the same feature with the fungal population, especially in the root compartment, and also slight differences in the root adherent soil. So please, next. We employed co-inertia analysis to evaluate the correlation between the two matrices, one representing, as we can see here, the abundance of OTUs, and the other one, the concentration of metabolites in each line. And this analysis revealed the correlation and covariation between these two data sets, which is influenced by the aggregation ratio. And we can see here that both OTUs, for example, here, and metabolite were co-positioned within the same spatial context for the same line. So that indicates a strong connection between specific OTUs and metabolites within each lines, implying a profound interrelationship between them. This was observed for roots and also for the root adherent soils. I don't know if you could follow me with these figures. Next, please, Christine. So we can see the same thing for the root adherent soil, where we have this separation between low aggregated lines and high aggregated lines. And this compositions of metabarcodine and metabolomic. So just to conclude with this first part, so the rhizosheet formation is under complex genetic control in pearl millets, and the data showed that it is mainly regulated by root exudation and rhizodeposition, and also that this rhizosheet is a heritable trait in pearl millets, and it correlates with changes in the rhizosphere and root exudase microbiota structure and root exudase composition. And Christine will tell us whether there is differences in carbon storage between these contrasted inbred lines.
Thank you, Wafa. So now I'm back to soy as Wafa, and I will try to explain you what we do with the isotopic measurements to provide some assessment of the agronomical practices. So let's come back to the concept of carbon storage and carbon sequestration. Here you have a view of one can be the organomineral interaction. This view is pretty old, in 2007, but and it changed a little bit since that time. But I think it's pretty clear to understand the process. On the left side here, that's as a line in brown, is a clay. And here you have all of the organic matter you can find in the soil. So they are divided into 3 different zones. One kinetic zone, where there is a lot of exchange, new carbon going to the kinetic zone, and the respiration come out from the kinetic zone. And you have the contact zone, very close to the clay, where the organic matter with strong integration and strong interaction remain next to the clay. So if we look at another consideration, if we look at the mean age of the carbon within these organic compounds, we can say that in the kinetic zone, we have carbons that will stay only very short time, maybe a year, less than a year, or several years. And at the opposite, next to the clay, it can remain very long time, more than 100 years. And in between, in the hydrophobic zone, we can have some carbon that is here since some decades. Because of this difference in time, that's also the difference in the concept of carbon storage. So carbon storage, it's only new carbon into the soil, but we don't know how many times it really stay into the soil. And there is a big difference when we talk about carbon sequestration, because in this concept, we want the carbon to stay into the soil and to really act as a mitigation way for the climate warming. And because I'm a isotopic girl, I will talk about isotopes as well. So I can change a little bit the notion of carbon age by using the C14. Here you have a view of the change of the C14 content in the atmosphere, and during a natural time without so many anthropogenic impacts, and for the BIAM peak. So for the carbon, old carbon, we have a blue point, and thereafter, by looking at carbon of some decades, we have red or purple points. And so the green to brown bar I had previously can be also shown as the blue-red bars here for the C40. So now, what about the heterotrophic respiration, a natural process, and the priming effect, a natural process also? So if you are in a steady state context, the amount of carbon that you will newly input into the soil will equals the soil carbon you will lose by heterotrophic respiration. Everything acts into the kinetic zones, and it's perfect. So, in contrary, if you have some deleterious practices like deep plowing, like no cover crops or something like that, the amount of carbon that you will input will be lower than organic carbon from soil that you lose during the same year. And in this condition, it's not only in the kinetic zones that the exchange occurs, but it also attacks the hydrophobic interaction zone and even the contact zone. So a good idea would be to input new carbon into the soil and so to increase by bringing more carbon to the soil, to increase the carbon stock, to keep it constant or even to increase it. But what we have to keep in mind is this new carbon can be very rich in energy and can lead to an increase in the priming effects. And in this case, there is big exchange into the kinetic zone, a lot of carbon new with a lot of energy. And there is a contraction of hydrophobic interaction. And the heterotrophic respiration appears here in the hydrophobic interaction. So there is a change in the molecular cortege, and finally less sequestrated carbon, because I will have more carbon of rapid turnover time than molecules with low turnover time. The question is, how can we assess the effects of this good practice? Because on the long term, the priming effects can lead finally to a decrease in the total organic carbon into the soil, or lead to an increase in increase of soil carbon. So the idea here is it's not possible only to look at the carbon content, because the change is very teeny, and we will have to wait for a very, very long time to have something very visible. So a way to do it is to follow the approach proposed by Jérôme Balédan in the 80s, is to use the natural labeling in C13, and why not in nitrogen 15, to make a difference between leguminous and non-leguminous. And so to be able to account for the new carbon and the old carbon into the soil, into the new soil organic storage. And what we propose here is to complete this approach by providing information on the nature of the lost carbon. Remember, I changed previously the green to brown bar into a blue-red bar. But now, with the priming, with the enhancement of the priming effect, I hacked on the hydrophobic interaction zone. So I removed some decades carbon, but some decades carbon knew the bond peak. So the red part of the blue to red bars is greatly reduced. And so we will have a big change in the C14 content into the soil organic carbon associated to an enhancement of the priming effect. And that's what we propose here to decide and to help in deciding if the idea, it's a good idea or a false good idea. This is an example we had together with Wafa and Thierry, and it was in the context of a DRF impulsion project. So we wanted to assess if the higher Rhizodeposition effectively provide more carbon on the long term or less carbon in soil on the long term. So we had C4 plants, C3 soil, and we measure carbon content, C13, C14, and of course, root adhering soil. And we wanted to evaluate what was the practice. Here has the same line that Wafa previously showed. So the L3, it was an intermediary, and the L132. And we were able to evaluate the amount of the new carbon and the amount of the lost carbon. And when we looked at all of the results, we definitively can definitively say that this line is very good because it's good. It's a good candidate for the 4 per mil initiative that intends to increase the stock of carbon into the soil, because the lost carbon is very low by comparison to this one. So yeah, priming effect at LSCE is based on instruments that are included into the Panoply platform, and you can have some slide here, some picture. Thierry?
Yes, thank you, Wafa. Thank you, Christine. Hi, everybody. So, as some slide as a conclusion. So, what is well known in the literature is this part of the carbon cycle, and starting with plant residues, including root in the soil, which with the decomposition and due to the remarkable activity of these residues and the mineralization. Christine, if you can follow my talk with your, yes, it's not so, and the result is the production of CO2. So, and this mineralization also contributes to the accumulation of microbial biomass, and the decomposition of these residues lead to the degraded organic matter, and at the end to organo-mineral assemblage, ending by stabilization of organic matter. So this is the well-known part of the carbon cycle. And next, the second part is well known, but in the different models, this part is more difficult to translate in the models. So you have the CO2 absorption by the plant due to photosynthesis, translocations of carbon into the roots and then into the soil. And this is the Rhizodeposition, including the root exudation. And most of these root exudates are organic compounds, such as sugars or organic acids. And what we have demonstrated since several years, and Wafa demonstrates this part, the sugars from the root exudation is transformed by due to the microbial synthesis into exopolysaccharides EPS. And so this EPS can be also associated with mineral and to leading to the stabilization of organic matter. And if next, yes, and this is this part of the carbon cycle was mentioned by Christine. Some of these organic sugars from root exudates contribute to the priming effect, consisting of the degradation of organic matter and the production of CO2. At the end, this, if you have the complete carbon cycle, is a simplified carbon cycle, but you have the two parts, the carbon storage on the right and the carbon sequestration on the left. Sequestration because this part is due to the sequestration of CO2 due to the photosynthesis of the plant. So next, I found this paper, including one of your colleagues of LSCE, Philippe Sier, on the microbial use efficiency, and I think that Daniel Goll will speak about this part. It was one of the research was mentioned yesterday. Next. Yes, in this paper, they try to compare two contrasting pathways in determining the relationship between microbial carbon use efficiency and the soil organic carbon storage. The first one on the top indicates that higher carbon use efficiency favors the accumulation of carbon storage through the increase of microbial biomass and byproducts. What we mentioned in this model, byproducts for us is something like exopolysaccharides, mentioned in my carbon cycle before. So it's a way to speak about the transformation of the root exudates by the bacteria and into bioproducts and especially exopolysaccharide. And the second pathway emphasizes the high carbon use efficiency simulates the soil organic carbon losses via an increase of the microbial biomass and the production of extracellular enzymes for the degradation. So in a sense, the two pathways, this one corresponds to the sequestration on the top to the sequestration of carbon, and the second one to the carbon storage and with the degradation of plant residues in soil. So at yes. So the conclusion, and we agree with this conclusion, they find that the carbon use efficiency is 4 times as important or as other evaluated factors. So this is a very important conclusion. The carbon use efficiency is the microbial carbon use efficiency is more important than the carbon input itself, the decomposition of the vertical transport in determining the carbon storage in the soil. And in addition, this microbial carbon use efficiency shows a positive correlation with the soil carbon contents. And this microbial carbon use efficiency is a major determinant of global carbon storage in soil. So this is one of the conclusions. Coming back to the previous question given by Wafa, is it possible to select plant genotype, plant varieties to increase the carbon storage and the carbon sequestration? Yes, the answer is yes. Yeah, and because you can see that if you increase the rhizodeposition and you increase the activity of bacteria able to transform the root exudates, which are sugars, a very labile carbon, into a carbon more stable. And at the end, the association with minerals can contribute to the stabilization of this carbon into soils. So thank you very much.
Okay, thank you very much. We can have questions for the two talks. Any questions?
Well, I have a question.
Maybe the talk of Daniel.
Oh, the talk of Daniel is cancelled because of health issues. So, but the presentations of Daniel and Eric Violet are on the cloud.
Okay.
Yeah, so this is the end of the talks for the soil session. So we have some discussion for those talks now, and then we can have a more general discussion too, if there are any other questions people would like to, or comments people would like to make. And so, Yoha, you had a
Yeah, I had a question about the meter project. Is still running, or will you still have a phase 2 or 3, or I don't know?
Maybe Nathalie wants to
Yeah.
answer.
But yeah, we already applied for the ANR project with this one, but we are not succeed. So we're still running with some budget from the Demeter 1, and we apply for a BP. We will try to apply for a BP project this year.
Okay. So the institute you mentioned about in Japan, they set up a local institute in Fukushima. Is that institute open also for kind of fund for international collaborative
Yes.
or only for local research? That institute.
They open different types of course, and
Yeah.
I think they have the intention to open it internationally, but so far I think it's money.
For money for the local.
Yeah. Or you should have a contact person
Yeah.
on site.
Thank you.
I have a question. So I have a question for the rhizodeposition. So I think there's a project in the US to I think increase suberin production in roots to store carbon. Is that something that would work? Is suberin will that be converted into a long-term carbon store, or is it more risky because it might cause degradation of the soil quality?
Yes, I know this work in US, and the selection of plant with higher content in suberin. If I remember well, my opinion at the end of the paper is something risky. It's not comparable with what we think about the transformation of root exudates, which is a very labile carbon into polysaccharide, because this is a stabilization, not for long term, but a stabilization of carbon. And if this carbon is associated with minerals, you can stabilize this. The suberin needs to be transformed in something else, and I was not completely convinced at the end of the paper. Yeah, but it is open to discussion for sure.
So Thierry, can you hear me?
Yes.
Okay. So in addition to the suberin, it's a lipid polymer, but I also worked when I was postdoc on this lipid secretion, like in the form of monoacylglycerols as a general lipid. They also secrete the roots, because I think most of your work you look only into the lipophilic, the hydrophilic compounds. Are you also interested in the hydrophobic lipid type of secretion in the extruded? It's another part of component.
I
Yes.
think that within this
Yes.
project, the problem is mostly related with suberin is not alone. And because there is some energetic components associated to suberin, like amino acid and especially sugars, it will at the end reduce the hydrophobic zone, even if the suberin will be in the hydrophobic zone. So maybe you will increase a little bit the amount of suberin into the soil, but you will decrease the other type of molecules there. So I'm not totally convinced by that. I think that it's really important. And something I forgot to mention, and it was repeated and repeated by Jérôme for a long time, is there is no action if you don't consider the soil history. And you cannot consider that one action in one soil under one type of climate can be reproduced everywhere and any time on any type of soil and on any type of soil, considering any type of practice before. So that this kind of study only look at one specific case, but the conclusion is always generic, and I think it's something very dangerous.
You speak about the work on suberin.
Yeah, for example, but there is some others like that, yeah.
I was afraid
I was actually thinking about the other components, not suberin. Suberin, I know, is protective. It normally sticks to the roots. It has some physiological functions. I was talking about the other part that's completely shed off by the plants. Inside there's not only sugars, metabolites, but also lipid components like mats and bags and all the things. So I'm sure maybe this part will be has less impact on the plant development or root function.
Yes, you are right. You are completely right. The first answer is more the reason why we focus on sugars is for the bacteria is carbon and energy sources, which is completely different compared to the other compounds. But you are right. The idea is not only to increase the carbon the exudation of sugars by the plant, if the selection is possible, if the selection is acceptable by the farmer. But we know that this is not only a question of quantity of residue deposits of carbon. It's also a question of quality. We have in the paper mentioned by Wafa, we have some indications that the quality of the root exudates is really important. And if we think about the total rhizodeposition, including the compounds we are speaking about, and we completely agree that what we have to look at is the composition of the root exudates, not only sugars, but probably other compounds, and the approach mentioned by Wafa did not exclude the different molecules. Even if with this technique the lipids are more difficult to identify, but we are right. We have to extend the analysis of root exudates beyond the only analysis of the sugars. I completely agree, but it's more difficult.
Yeah, we also know that plants produce some fatty acids to attract some gram-positive bacteria. So there are different compounds and they are all important in the recruitment of the microbiota associated with the plant. But we focus more on this exopolysaccharide because we have been doing this for many years ago. And we showed that bacteria that produce this exopolysaccharide increase the size of this rhizosheet. And also it allows the plant to tolerate water stress because we used also pollutants that don't produce this polysaccharide. And we could see that the size of this rhizosheet was mainly associated with the bacterial polysaccharides. So we have some evidence on this, and we have also some evidences that when this rhizosheet, its size, which is when it is increased, we have a better uptake of water and nutrients by the plant. So it is a way also to for the plant to uptake water when there is water stress, because these polymers are known to retain water. Yeah. So we have some evidences because we used some isogenic mutants that produce or doesn't produce or don't produce exopolysaccharide, but all the other compounds, they are important. Plants use different compounds to attract different bacterial population, which play important role in the development and also health of the plant.
To go further in the collaboration between LSCE with Christine and with Lemire, I think that we have also to involve Daniel Gall. We discussed about this with Christine about the microbial use efficiency, carbon use efficiency, because this point is in the different models. This part is not well described and quantified. So maybe in the future, I don't know if Christine agree with that. We have to increase the collaboration of with modelization with this part of the model, which is not as well known as the mineralization, because for the agronomists, the carbon cycle is somewhere resumed to the carbon decomposition, and we are in our work speaking about the carbon elaboration through the polysaccharides. So this is one proposal to go further in the collaboration with LSCE. This is not the end of the story.
Okay, thanks, Thierry. So do we have any more questions on this soil session? Online as well? So if we don't have any, there's a question. So it's a question for the Demeter project. So from the farmer's point of view, with a strategy that is not targeting soil decontamination, is the exposure to cesium now within acceptable limits to work in the fields? Or will the farmers have to use special equipment to limit their exposure to cesium dust in the long term?
Yeah, but that's the question. If you don't decontaminate the soils and you plant these safe plants, it's so it depends on the place. But then you would imagine that farmers would only use their machinery during limited times, so the time of exposition would remain limited. I think that's the idea. But then it depends, of course, on the fallout level, depending on the place. But I would say time of exposition is limiting as much as possible. The time of exposition would be the.
And so is that being done? Is that being done now? So people are farming on.
But
soil.
most of the areas have been decontaminated, but there is at least in one village, and I guess this happens in other villages, there are places where people don't want their soils to be, because they say that's their territory and then you can't just remove soil like that. So they refuse to do that and they implement other practices. So typically there you could test that.